Hello students, welcome back to the channel. In this video, we are going to study two type of active transport. One is primary active transport, another is secondary active transport. Primary active transport will use ATP directly. Means directly they are doing hydrolysis of ATP to move the ions. For example, in this diagram, if you see here, outside the cell condition and inside the cell, this is sodium potassium exchange pump. We are making use of the sodium potassium exchange pump. Clear? And if you see here, three sodium ions are binding and then ATP is binding. ATP hydrolysis will bring the change in the protein structure. Okay, see, it will bring the change in the protein structure so that this gate will open. See, this gate was closed, now this gate is open and sodium will be going out. First thing. Second, if you see the diagram very carefully, you will find here the potassium was not able to bind, but now gates are open in such a way, means conformational change in the protein in such a way, now potassium is able to bind. So binding of the potassium is going on, and after this phosphate ions will be released, which will bring the protein back to its normal shape. Okay, means potassium are liberated and these sites are open to again make the sodium bind. So here if you see in this diagram, direct hydrolysis of ATP is there, which is providing the energy for the transport. Comfortable? Now see next. Secondary active transport. In this, see one thing, this is a glucose molecule. This glucose molecule is moving from low concentration, from its low concentration to its high concentration. Is it clear? This is a diagram we are taking of one villi. You know very well villi we study in the small intestine. So villi, epithelial, one cell. This is brush bordered epithelium. Okay. And we are finding the glucose is moving against its concentration gradient, but with the help of sodium, because here sodium is moving along the concentration gradient. Means sodium is moving from its high concentration to its low concentration, and it is giving the glucose to move from low concentration to high concentration. But how? Here I'm not using ATP. You check my diagram carefully. Here ATP usage is not there. So here what we are doing, first of all, we have to create inside the villi low sodium. So we are actually using sodium potassium exchange pump in which we are pumping 3 sodium out. And because we are pumping 3 sodium out, the sodium inside is becoming low. So sodium is moving from its high concentration to its low concentration, and it is helping the glucose to move against its concentration gradient. Is it clear? So if you see sodium potassium exchange pump will establish a concentration gradient. Now ion want to diffuse back, and this will provide energy for the glucose to come against its concentration gradient. So generally it is used with the help of one protein only, which move both the ions and actively transport molecule across the membrane. Now here if you see both molecules are moving in the same direction, we are saying this is symport condition. Okay, and sometimes the molecules may move in opposite direction. We will say antiport condition. So here glucose is going against the concentration gradient, but directly ATP usage is not there. It is indirectly ATP usage. That is why we say it is a secondary active transport. The movement of substance against its concentration gradient is accomplished by using energy by letting ions move down their concentration gradient. Please understand this line very carefully. Ki sodium is moving along its gradient, making the glucose move against its gradient. Is it clear? I hope so. This video might be helpful for you. Thanks for watching.